Biologists investigating the morphologic changes associated with reversible cellular injury perform a procedure on anesthetized mice to assess the effects of transient hepatic ischemia. During the experiment, they clamp the hepatic artery and obtain liver biopsy samples at varying intervals. The samples are then examined by electron microscopy. Cells that are exposed to longer ischemic periods are found to have reduced numbers of ribosomes attached to the endoplasmic reticulum. The question is, this structural change is most likely to impair which of the following cellular functions? Your options are A, ATP production, B, drug detoxification, C, synthesis of cell membrane proteins, D, synthesis of cytosolic proteins, E, synthesis of steroid hormones. Type your answer in the comment box. Congratulations if your answer is C, which is synthesis of cell membrane proteins. Ribosomes are cellular organelles that synthesize proteins. Each ribosome consists of two subunits. The small ribosomal 40S subunit is responsible for binding mRNA, the protein synthesis template, and tRNA, that carries amino acids. The larger 60S subunit contains peptidyltransferase, the enzyme that catalyzes peptide bond formation between amino acids. All ribosomes begin protein translation in the cytoplasm, but some translocate to the rough endoplasmic reticulum during protein synthesis, depending on the protein's target destination. Free ribosomes remain floating in the cytosol throughout protein synthesis. They are responsible for translating proteins found within the cytosol, nucleosol, peroxisome matrix, and nuclear-encoded mitochondrial proteins. Attached ribosomes bind to the rough endoplasmic reticulum after protein translation begins. They synthesize most secretory proteins, the integral membrane proteins of the nucleus and cell membrane, and proteins within the endoplasmic reticulum, Golgi network, and lysosomes. The rough endoplasmic reticulum is particularly well developed in protein-secreting cells like pancreatic and plasma cells. Ribosomes attach to the rough endoplasmic reticulum via the translocon, a protein complex containing ribophorins that bind the large 60S subunit. Proteins in both of these cellular compartments are synthesized by free ribosomes in the cytosol. If you chose any option other than C, you were incorrect. Let's briefly discuss the other options. Choices B and E, steroid hormone synthesis and drug detoxification, are performed by various proteins found within the smooth endoplasmic reticulum. The smooth endoplasmic reticulum does not bind to ribosomes as it lacks the translocon complex. If you're finding this difficult to understand, consider watching this episode again.